The next unit we're visiting is DNA structure. There's a lot of different parts of DNA that we're going to discover, but the very first part is what it is, what it's made of, how it is formed, and basically about the shape of DNA. So the structure is DNA is a nucleic acid. It's made of monomers called nucleotides. There's 3 parts of DNA. There's the 5-carbon sugar, and that's called deoxyribo. And then you have a phosphate group, and then you have a nitrogenous base. So that's what makes up the nucleotide. All right, in discussing the nitrogenous bases, they're basically paired to fit each other. Purines pair with pyrimidines. So the 2 nitrogenous bases that are purines are adenine and guanine. And if you look up here, you have adenosine and guanine. Those 2 are double ringed, and they cannot fit together. So purines have to be paired with a pyrimidine. Pyrimidines are single ringed, and they are thymine and cytosine. So how they're paired together is adenine pairs with thymine and cytosine pairs with guanine. Okay, and like I said before, purines cannot fit together because they're shaped so similarly that they are paired with a complementary pyrimidine. A pairs with T and C pairs with G. And so here's just a picture of the example. And this should be a review of your middle school. Now, these base pairs are called the Chargaff's rule. Let's watch a quick video about Chargaff.
The DNA molecule is made up of very long chains of the 4 bases, A, C, G, and T. In 1950, Erwin Chargaff published a paper stating that in DNA of any given species, the ratio of adenine to thymine is equal, as is the ratio of cytosine to guanine. This is known as Chargaff's ratios, and it was a crucial clue that helped solve the structure of DNA. Chargaff's ratios are universal. All forms of life obey this rule. Only the balance of AT pairs and CG pairs varies between species.
Okay, so Chargaff's rule basically explains how adenine and thymine and cytosine and guanine are paired together. The differences in how they're organized on the DNA strand is what makes us look differently. So this is the actual shape of DNA is called the double helix, where if you were to iron it flat, then you would see that you have phosphate, sugar, alternating down the sides. And then you have the nitrogenous bases bound together. And they're bound together by those hydrogen molecules. And we talked about hydrogen being so sticky that it's almost like a magnet. The 2 nitrogenous bases will be attracted to each other, but they can be easily broken apart also. And so that's why hydrogen is like the ultimate element for bonding. So we have the shape here, and then we have a flat version of the shape, and then this is the molecular version. If you look here, you see A and T meet in the middle, C and G, A, T, C, G, and that pattern is what determines the genetic variation. What I also want you to look at is that the phosphate is going up on this side, and on this side the phosphate is going down. So that's what gives the DNA its double helix shape. Now the hydrogen bonds are going to join the nitrogenous bases together really tightly, and they don't fit exactly, so that's what makes the molecule twist like that. RNA, on the other hand, is a disposable copy of DNA. It's single-stranded, and it has 3 parts also: sugar, phosphate, and nitrogenous base. Okay, the difference in RNA is that the nitrogenous bases, thymine, is not available. So the base pairs are adenine pairs with uracil, cytosine pairs with guanine. So replacing thymine with uracil. So in summary, DNA is located in the nucleus of a cell. All DNA hold instruction to make proteins. RNA is the disposable copy of DNA. And what RNA does is it takes information from the nucleus to the ribosome in order to make proteins. And we will talk about the total process of protein synthesis probably in the next video and in class.